Okay, thank you all so much for being here. I'll pass the webcam and microphone over to John Barlow, who's the chair of the evaluation committee for Connor's thesis defense, so we can go over some logistics.
Yeah, thanks for everybody for coming. I'll just sort of say that typical logistics for UVM thesis defense is we have a public thesis presentation by Connor with some time for questions afterwards from the public. Usually at that time, the committee doesn't really ask many questions or anything and just lets it open for public questions. And then we all ask people who are not on Connor's defense committee to leave the Zoom, and we meet with Connor privately to talk about the scope of his work. So that's the general logistics. And with that, I'll turn it back to Laurent, and he can do the more formal introduction for the student.
Thank you so much. So thank you, Dr. John Barlow. I also want to thank Dr. Nick Cheney for being with us today to complete the committee to evaluate Connor's thesis defense. I'm so happy that I'm talking first because I've seen Connor's slide, and Connor will start with a very Connor thing. So you will be thanking a lot of people early that helped him throughout his master's program. But make no mistake, this was all Connor's work. Connor did the thing that all great scientists do, especially in complex systems, and a system as complex as human health, took in a lot of expertise from other disciplines. You can't fully understand a complex systems or human health from just one perspective. So Connor has been, talking with experts and reading papers in ecology, network science, data science, infectious disease, microbiology, just an impressive piece of work to take in all of those ideas, all of those tools, and try to figure out what is the most useful to study patterns of co-infection. And I'm very, very impressed with this piece of work because Connor is a very creative person. And when you get exposed to a lot of ideas, you get so many projects. And the hardest thing to do in science, because a project is never done, is to figure out when to let it go. And I think Connor figure out the right balance. We have a lot of ideas for future work, but I think the piece of work you're about to see today is amazingly complete, tells a compelling scientific story, and is just a great master's thesis. So hopefully for all of us, this is a time for congratulations and no goodbyes, and we'll be able to continue working on some of Connor's ideas in the future. But I couldn't be more proud, and I want to also thank Dr. Russ Colgate, who's Connor's co-advisor. Couldn't be more proud to have our names on this piece of work. I think is just a really, really impressive master's thesis, and I'm excited for you all to hear about it today. So all that said, I'll turn it over to Connor to present his master's thesis entitled Network and Ecological Methods to Study Enteric Pathogens Co-Infection. Connor, the microphone is yours.
All right. Thank you very much. And thank you everybody for coming, and thank you for that introduction. As Laurent said, today we're going to be talking about network and ecological methods to study enteric pathogen co-infections. Now, before we begin, I want to take this opportunity to thank everybody that made this possible. First, I'd like to thank my advisors, Drs. Ross Colgate and Laurent Hubert-Dufresne. Their encouragement, their patience, and their guidance really allowed me to grow as a data science student and really helped shape this project into what it is today. I'd also like to thank my thesis committee members, Drs. John Barlow and Nicholas Cheney, for agreeing to be a part of this final stage of my graduate career. And for that, I thank you very much, and it means a lot. I'd like to thank the Laboratory for Structure and Dynamics, as well as the newly formed Complex Data Lab. I've had the honor of working with those individuals for 2 years, and they're a really talented and respectful group of individuals, and they have done and will continue to do great work. I'd also like to thank the Translational Global Infectious Disease Research Center, or TIGER, and the LCOM Vaccine Testing Center. I've had the pleasure of presenting this work to the TIGER group a couple times before, and the feedback that I've gotten from them has been instrumental in shaping this project into what it is today. I'd also like to thank the LCOM Vaccine Testing Center. For those of you that don't know, that was my home for 2.5 years before graduate school. And one could really not ask for a better environment in which to work. Just a really excellent group of individuals, and there's really not enough time to thank Beth and everyone that is part of that group for everything that they've done for me. I'd also like to thank Chris Houston, who took me on as an undergraduate back in 2014. And then he and Beth hired me as a technician after I graduated. And it was Chris who actually gave me my first data science project when I was first learning how to code in R. And that was the first initial push that led me down the road of data science to where I am today. I'd also like to thank David Dewhurst and Dominique Graval for helping us in developing this later chapter of this thesis. That work was really instrumental in shaping that. So without further ado, let's move on to the science. So the talk today is going to be presented into 4 different parts. First, we'll talk about diarrheal disease, its impact on global health, and then also co-infections in the context of diarrheal disease, and why it may be important to identify pathogen pairs. We'll talk about using ensembles of bipartite networks to identify pathogen pairs that appear more or less than what we would expect given their individual prevalence. And then we will use a joint species distribution model to characterize those pathogen pairs as a shared response to an environmental niche versus a possible correlation between pathogens that could represent an interaction between them. And then finally, we'll look at what do these 2 models tell us in tandem about how we can identify pathogen pairs for possible further investigation in order to identify superinfections in the context of diarrheal disease. So why diarrheal disease? Well, it's one of the causes of death globally, and it's a major burden on global health. Even though we've made great strides in the past 15 years, it still affects 1.7 billion people globally and can lead to the deaths of 525,000 children under 5 years of age. This is especially a problem in lower to middle income countries, where diarrheal disease becomes one of the leading causes of death. And the death rates due to diarrheal disease is negatively correlated with GDP. And diarrheal disease at a young age is especially critical because as young kids experience chronic early onset diarrhea, the stress that causes can lead to physical and mental stunting. So then as those young children develop and grow into working members of society, they're set at a disadvantage compared to societies that don't have that burden of diarrheal disease. And what happens is that because they're at a disadvantage, that can then predispose the subsequent generation to increased rates of diarrheal disease. And then they experience their own physical and mental stunting. And what happens is this problem is perpetuated over generations, and the effect of diarrheal disease on young kids can compound over time. So it's important to fight diarrheal disease, but it's important to attack it, especially in young kids, in order to break out of this cycle. And while there are a number of factors that are responsible for causing diarrhea, one of the main reasons is due to enteric pathogen infection. One of the leading causes of pathogen infection is rotavirus, followed by adenovirus, and then common bacterial infections are cholera, Shigella, and toxic variants of E. coli. So that'll be your enterotoxigenic E. coli, enteropathogenic, and enteroaggregative E. coli. And with this high prevalence of enteric pathogens in these populations, especially in some locations more than others, I think it would be reasonable to suspect that there's cases of pathogen co-infection. And there actually has been work looking into pathogen co-infection and what that means in the context of diarrheal disease. For example, the figure on the left looks at 199 cases of acute diarrhea in Chile. And the researchers find that in 27.1% of cases, participants had a polymicrobial infection, meaning more than one bacteria or pathogen was found. And in cases with that polymicrobial infection, those participants had increased frequencies of nausea and increased abdominal pain. And then in another study looking at 1,022 children in Southeast China, researchers found that in cases where rotavirus co-infected with another pathogen or either virus or bacteria, those participants had increased diarrheal severity. And so we can see here is that there is some evidence that perhaps co-infection can lead to an enhanced disease state compared to those with just a mono infection. So it's important to understand, well, what are these patterns of pathogen co-infection and what do they mean? And that's going to be the main question we're going to ask today. How do we identify these patterns and what do they mean? And in order to answer that question, we're going to ask two more questions, which is how do we find patterns of pathogen co-infection that appear outside of the prevalence of the individual prevalence of those two pathogens? And then we're going to characterize pathogen pairs based on whether they have a shared response to an environmental niche, or if there is evidence of a biotic interaction between those pathogens. So our first step is, how do we identify patterns of pathogen co-infection that occur outside what we would expect given the prevalence of those individual pathogens? And so to answer this question, we'll use two data sets. The first one is from the PROVIDE study, which is a collaboration between University of Vermont, University of Virginia, and the ICDDRB in Dhaka, Bangladesh. That study included 700 children, but we're only going to select 270 based on participants who had both diarrheal and asymptomatic stools, as well as corresponding qPCR data. And this was a study looking at the efficacy of polio and rotavirus vaccines in developing countries. Another very similar study that we're going to use is the MAL-ED study, which consists of 8 locations globally. And this looks at the effects of diarrheal disease on undernutrition in kids, and then the effect of diarrheal disease and undernutrition with vaccine efficacy. And because we want to compare the results from the two studies in this section, we're only going to look at the Dhaka, Bangladesh study site, which includes 210 children. And because, as I mentioned earlier, going after diarrheal disease is important at a young age, we're going to only look at the first 53 weeks of life, which is approximately the first year of life in these participants. So these data subsets that we use are going to consist of diarrheal or asymptomatic stools. So asymptomatic stools are stool collections that are during regular study visits, versus diarrheal stools are those collected during diarrheal episodes, which is defined as 3 or more loose stools in a 24-hour period, separated by a number of diarrheal-free days, depending on the study. And for each stool, we have qPCR or RT-PCR data, which is associated for each stool sample, measuring the amount of genetic material in those stool samples for 30 different pathogens. And we are going to say that a pathogen is present if that qPCR data or that CT value is less than 35. And the last thing I want to draw your attention to is there are a large number of asymptomatic stools compared to diarrheal stools, at least twice as many. And that's important. That's going to be important later on. So within these data subsets, we can see that some pathogens have a high individual prevalence compared to others. For example, EEC and ETEC appear much more than B. fragilis and Cryptosporidium. And so what we would expect is if we just took a brute force frequency count of the number of times two pathogens show up together, I think it would be reasonable to predict that two pathogens that occur individually in very high amounts will also appear in a lot of co-infections. But what we want to do is identify pathogen pairs that occur outside those individual frequencies. And so to do that, we'll use a bipartite network representation, which is a way to represent the data in two groups, in this case, stool samples and pathogens. And we will draw an edge between stool sample I and pathogen J if pathogen J is found in stool sample I. And that'll be defined as having a CT or CQ value less than 35. And the reason we do this is qPCR, the test that's the gold standard during the COVID-19 pandemic, is highly sensitive and highly specific. And so that's why we're going to have this threshold value of less than 35. And another way to represent a bipartite network is via a one-mode projection, where we take all the members of one group and we draw an edge between those members if they share a neighbor from the opposite group. So in this case, we'll take all the pathogens from the bipartite network and we'll draw an edge between them if they share a stool sample. And that edge will be weighted by the number of stool samples that they share. And so this is a way for us to look at the bipartite network and count the number of co-infections we find in stool samples. And what we'll do is we'll then take that original bipartite network and we'll randomize it. We'll rewire the edges between those nodes in a way that preserves the network structure. So we're going to preserve the degree distribution for both the stool samples and the pathogens. And then in that random realization of the original bipartite graph, we're going to look at the one mode projection to count the number of co-infections in that random network. We're going to rewire those edges 15,000 times to ensure that more likely than not, we are going to rewire every edge, and we're going to repeat that for 10,000 copies of the 10,000 random representations of that graph. And then what that'll do is that'll create a distribution of the number of co-occurrences between two pathogens. And that distribution is representative of the number of times that we would expect two pathogens to occur, given the structure of the network or given their individual prevalence. And so what we can see here in the figure on the right is this is the distribution given that network structure. These solid lines represent the actual number of times two pathogens co-occur. These dotted lines and these dashed lines represent the bounds that contain 95% and 99% of that random ensemble respectively. And then values along the x-axis here represent standard deviations from the mean. And a negative value is in place in front of that standard deviation if it occurs below the mean of that ensemble. And so for every pathogen pair, we'll assign a percentile value, which is the percent of the random ensemble that is less than the observed number of co-occurrences. And then the second metric we'll use is the discordance value, which is the distance from the percentile of the theoretical mean, which will be 0.5, and the actual observed percentile, which is the where that observed number of co-occurrences falls in that ensemble. And the idea here is that a larger discordance value means that the observed number of co-occurrences fall far outside of what we would expect given the individual prevalences of those pathogens. And so what we'll do is we will rank those pathogen pairs within each stool type based on the average discordance between two studies, meaning that two pathogen pair that appears more or less than what we'd expect given their individual prevalence in both studies will have a higher rank compared to those that don't. We'll partition the data set into 4 different subsets based on study and stool type. And when we look at the top 12 pairs ranked by their average discordance found in asymptomatic stools, we find that 7 out of those 12 pairs are bacterial co-infections, and in all but one case appear more than what we would expect given their individual prevalence. We find that 5 out of those 12 pairs are bacterial virus co-infections, and in all but 2 cases appear less than what we'd expect given their individual prevalence. When we do the same for diarrheal stools, we find that 5 out of those 12 pairs are bacterial co-infections, and in all cases appear higher than what we'd expect given their individual prevalence. And 5 out of those 12 are bacterial virus pairs, and in all but 1 case appear less than what we would expect. In the diarrheal stools, we also have 1 case of a virus-virus co-infection and 1 case of a bacterial protist infection. So what we'll do is we'll take those top 24 pathogen pairs, and we find that 4 of those pairs are shared between the asymptomatic and diarrheal stools. And all of those pairs contain enterotoxigenic E. coli, or ETEC, with EPEC, which is enteropathogenic E. coli, Campylobacter, enteroaggregative E. coli, or rotavirus. So far we've identified these pathogen pairs that appear more or less than what we'd expect given their individual prevalence. But our next question is well, what effect do these pathogen pairs have, both on the burden of diarrhea and on the participants themselves in which these pathogen pairs are found? And so we'll take these 20 distinct pairs and we'll look at, for all the stool samples where both these pathogens are found together, what is the proportion of these stools that are diarrheal versus asymptomatic? And we find that in 4 of those bacteria pairs, over 50% of them are from diarrheal stools. And in bacteria virus pairs, we find that 7 of those pairs have over 50% diarrheal, which means that more likely than not, where those pathogens are found, those will be from a diarrheal stool. And we can, especially in the case of bacteria viruses, where the majority of those bacteria viruses pairs have a stronger preference towards diarrheal stools. The other thing I want to point out is that when we look at the bacteria virus pairs and we rank them by the proportion of their diarrheal stools compared to asymptomatic, the top 4 all contain rotavirus. And these are especially meaningful results because if you remember from previous slides, there was a much larger number of asymptomatic stools compared to diarrheal. So if whether that stool is diarrheal or asymptomatic is independent of the pathogen pairs found in there, we would expect that same relationship to hold. But we don't see that. For a lot of these pathogen pairs, they are more diarrheal, even though that is the minority class in that data set. And so what we've been looking at now in the configuration model and in these figures is counting up the stools individually, looking at those stools independent of each other. But then the next question is okay, well, in cases where these co-infections happen, what does that mean for the individual? What does that mean for the participant? So to do that, we'll classify participants into 1 of 4 categories based on their exposure to either both or one of the pathogens found in these pairs, and whether they experience those pathogens at the same time or not. We can only consider a subset of these participants because some participants had recorded diarrheal episodes, but no corresponding qPCR data, so we cannot definitively put them in one class or another. But for those that remain, we are going to compare the average days of diarrhea between those 4 groups. And the idea there is that individuals that have more days of diarrhea can be considered they're more sick than those that do not. And so that will be our proxy for the health of the individual. And when we compare those average days of diarrhea among those 4 groups, we see that 75% of those pairs had higher days of diarrhea on average in cases where there was concurrent co-infection of those pathogens. We find that 4 bacteria pairs had the highest days of diarrhea in concurrent co-infection. And for the bacteria virus pairs, especially in the case of the PROVIDE study, all of those pathogen pairs, the highest days of diarrhea was either found, the majority of cases was with concurrent co-infection, or where the individual experienced both pathogens, but not at the same time. And there is a discrepancy there in the significance between the MALI-D and the PROVIDE data sets. But in cases where there is a difference between the groups, that relationship still holds, where the more days of diarrhea are either found in the majority of cases in concurrent infection in MALI-D, or where they experience both pathogens, just not at the same time. So in conclusion with this, we can identify pathogen pairs that occur more or less than what we would expect given their individual prevalence. We find that pair composition affects whether the majority of those co-infections were found in diarrheal versus asymptomatic stools, and the impact on the individual as measured by days of diarrhea on average among those 4 groups. And we find that those with bacterial virus infection have more diarrheal samples compared to asymptomatic, and have more days of diarrhea compared to those that don't. And what does that really mean when we find a pathogen pair that appears more or less than what we'd expect? Well, one example we can give is a possible mechanistic reason. And in the case of B. fragilis, or Bacteroides fragilis, and E. coli, this is actually a synergy that's been studied extensively, especially in the 1980s. And while I would say that the results in vivo are a little more inconclusive, in vitro, it has been hypothesized that they could work together by inhibiting neutrophil phagocytosis, so inhibiting the immune system, or increasing abscess formation, which allows for increased invasion. And so this, because there are molecular mechanisms that are working in tandem, I would define this as a mechanistic reason for why 2 pathogens may work together. And that's why we would expect to find them more than we would expect, together more than we would expect given their individual prevalence. And then another reason could be due to an environmental niche. Now let's take the case of rotavirus and ETEC. Now, rotavirus and ETEC were found in this case less than we would expect given their individual prevalence. And what we know from rotavirus is that is more associated with the dry season, which are the colder months in Dhaka, Bangladesh, versus ETEC is more associated with the warmer temperatures, which would be the wet season in Dhaka, Bangladesh. And so what we have is two pathogens that have opposing responses to environmental niches, where one favors the wet season, one favors the dry season. And so that could explain why we see pathogens that appear less than what we would expect, or appear together less than what we would expect, because they appear at different times of the year. And that actually brings me to a limitation in the configuration model, is that we do not incorporate a lot of these factors that we know are responsible for or could contribute to pathogen infection. Things like time, whether it's calendar or age, the location where these stool samples were collected, as well as socioeconomic status, access to clean drinking water, and proper sanitation. Now, the configuration model does not incorporate this information. So then the next question is, well, how are these pathogens that we see more or less than we would expect, how much of that is due to a shared favorable environmental condition or shared response to an environmental niche versus a potential mechanistic biotic relationship between those two pathogens? And so what we're going to say, our next question is going to be, what pathogen pairs are the response or the result of an interaction between them versus a shared response to the similar or opposing environmental conditions, like what we see with rotavirus and ETEC, for example. And so to answer this question and incorporate the environment, we're going to use a multiple multivariate regression with an error term to compare that response to the environment versus a potential biotic relationship between pathogens. And what that model looks like is it contains three parts. The first part here is a presence-absence matrix, where the number of columns equal pathogens and the number of rows is equal to participants. And a 1 is in a cell if that pathogen was found in that participant. And that'll be the output for this model. The input is going to consist of our environmental controls, which contain our beta coefficients, which for those of you that are familiar with regression is the relationship between the input data X and the output data, which would be our presence-absence matrix. And this will represent our environmental influence on the presence of pathogens in different participants. The third portion is a residual error term. And what that means is that when we control for the environment using these regression coefficients, whatever is left over and whatever predictive power is needed to predict those pathogens that's not accounted for in the environment is going to be captured in that residual error. And that error is sampled from a multivariate normal distribution. And that multivariate normal is going to be shaped by a correlation matrix R. And what that means is that for two pathogens that are positively correlated, that multivariate normal distribution is going to be skewed along the two dimensions representing those pathogens in the positive direction, so that when we sample from that distribution, if one pathogen is found, more likely than not, the other pathogen will also be found or has an increased chance of being found. And so to use this model, we're going to only use the MAL-ED study, but we're going to expand out to all 8 locations in order to capture that location variable. And we are going to partition the data set based on the age of the age of the participant. So we'll have a newborn model, which is 0 to 9 months, and an infant model, which is 9 to 15 months. Now, the reason we do that is infants undergo a major dietary change where they go from breastfeeding to non-breastfeeding. So in that case, nutritional information might be inappropriate for stool samples that were collected from infants that were still breastfeeding. And so we'll have the newborn model and the infant model, and we'll also expand that infant model out to 15 months in order to capture some of that nutritional information, things like dairy count, legume count, and so on. And so what we'll also do is from the MAL-ED data set, it's a huge data set. We'll have to eliminate some variables. Some variables such as, was this assay run, yes or no, are eliminated. And then also variables where they had sparse sampling. So for example, blood draws done only at 7 and 15 months respectively. we did some form of imputation, it would be inappropriate to do a last observation carried forward across that many observations or that long a time. But even with those variables removed, we still have 69 abiotic factors included in the newborn model and 134 in the infant model. And so if we added all of those to the JSDM, that would still be a very complicated, very complex model that might not even converge when we sample the posterior and would be very hard to interpret. So what we're going to do is we're going to use a forward stepwise selection using a generalized linear mix model with a probit link. And for every pathogen, we're going to select the best model that predicts whether that pathogen was found or not in that sample. And then we're going to take those selected models for all those pathogens, and we're going to look at the abiotic factors or predictors that were included in 2 or more models. And so when we count the number of models where these abiotic factors were included, or number of pathogens where these models were needed, what we see is that one of the most common abiotic predictors is to do with location and time, whether it's the age of the individual or calendar time represented by the wet season. And so we'll take all these abiotic predictors that were included in 2 or more models, and we'll use this as our set of abiotic factors to represent our environmental response or environmental influence in the model. And so to do this, we'll create 4 model variants, which is going to be the null model, which controls for prevalence only, the community only variant, which contains only the residual correlations between pathogens, the environment only, which contains only those abiotic predictors we have selected for previously, the full variant, which contains both the environment information and the residual correlations between pathogens. Now, for all these variants in both models, we're going to use a Gibbs sampler to sample from the posterior. We'll run 4 chains to check for convergence. And then we will only use 75% of that data as training data to sample this posterior. And then within that training data, we'll use 10-fold cross-validation to see how stable the DIC score is in a way to see how well those models do on different partitions of that training data. And so a deviance information criterion is a generalization of the Akaki information criterion that essentially grades the variant based on how well it predicts the co-occurrence of pathogens given its complexity. So simpler, more accurate models are going to have a lower DIC score and therefore are better models compared to larger, less accurate models. And so what we see is that the variants that incorporate environment information, the environment and the full variant, do much better as measured by a DIC score compared to those that don't incorporate the environmental information. And those that incorporate that residual correlation between pathogens, which would be comparing the null variant to the community variant and comparing the environment variant to the full variant, there's only a marginal increase in performance when we include those residual correlations between pathogens. And so what we see is that the environment carries a lot of the weight when improving these models. Now, before we move on, I want to take a minute to describe the JSDM as a junk drawer. Now, when we have that residual error term and those correlations between pathogens, when we look just at that, we don't exactly know what is driving those correlations between pathogens. And similar to a junk drawer, we don't really know what's in there until we start pulling items out of the drawer in order to organize it. And only then can we start to characterize and understand what the makeup is of that drawer. And the residual error works the same way, where as we start to pull out abiotic predictors, we can start to organize that signal in the residual error. And what we may see as correlations between pathogens may just be a shared response to the environment. And we can actually see that here when we look at the posterior distribution of those residual correlations between pathogens. And so what we see here on the left is the community-only variant, and on the right is the residual correlations found in the full variant. And when an edge is drawn between pathogens or a tile is filled in with a value, if 90% of the higher density interval or 95% of that posterior distribution does not contain 0, which means most of that posterior is either all positive or all negative. And what this means is that when we add in the environment information here by shifting over to the full variant, what we see is that a lot of those positive correlations, the HDI shifts towards 0. That means that those correlations become weaker in the context of the environment. We see the same thing with the infant model, where we see positive and negative correlations in the community-only variant. But when we start to incorporate environmental information, those correlations, the strength of those correlations reduce and move towards 0 and go away. We see a different pattern when we look at the correlation between environmental predictors between pathogens. So this is our shared response to the environment between pathogens. And what we see is when we compare the environment-only variant on the left to the full variant on the right, we see that that shared environmental response does not really change when we incorporate that residual error and that correlation between pathogens, which means that if we go back to the junk drawer analogy, we've already have those items in hand. So when we add in the unknown, that's not going to change what we already have in hand. And so we see that the response to the environment is fairly robust, even when we add in the residual correlations between pathogens or those biotic relationships. We see the same thing in the infant model, where again, that shared response to the environment remains the same, even when we add in those biotic relationships. And so what we'll do is we'll take the full model from both the newborn model on the left and the infant model on the right, and we'll compare the full variant. And what these plots are showing is within that full variant, what is the strength of the shared response to the environment represented along the horizontal axis versus residual correlations between pathogens, which is the strength of those biotic relationships. And each dot represents a pathogen pair, and that is colored based on whether the environment correlation or the residual correlation, that HDI did not contain 0, right? And so what we see is that for these pathogen pairs, they are spread more along the horizontal axis, which means that the strength of that shared response to the environment is much stronger than the residual correlation between pathogens. And this fits to what we see in the DIC score, where models that incorporate that environment information do much better. And then we see that in the correlation plots shown previously, where the residual correlation changes in response to the environment, but the environment response does not change when we add in those residual correlations. And so what we can see here is that when we look at the posterior that's sampled using the training data, we see that the shared response to the environment is much stronger than the residual correlations between pathogens. And so next what we'll do is we'll look at how well these models do in an out of sample context. So how well is their testing accuracy? So you remember we reserved 75% of the data for training, and then we have 25% for testing. And what we'll do is we'll use that testing data as well as the posterior distribution of those parameters that we were just looking at previously in order to generate a posterior predictive distribution, which is the estimated number of co-occurrences that the models will give based on what we observe in that testing data. And then we will look at the actual number of co-occurrences in that testing data, and we will compare where the actual number of co-occurrences falls compared to that posterior predictive distribution. And we'll use a binomial test where the p-value will tell us how far outside or inside that actual number of co-occurrences falls within the posterior predictive distribution. And the idea here is that if a p-value is less than 0.05, then that observed number falls outside that distribution. And we can say that that model does not adequately predict the co-occurrence of those two pathogens. Versus if the p-value is greater than 0.05, we can say that that model does adequately approximate those co-occurrences. And that'll generate these figures here, where we can visualize all the pathogen pairs. Tiles are shaded gray if the median of the posterior predictive distribution or the actual number of co-occurrences within the test data set occurred less than 5 times. and they are colored yellow if that p-value is less than 0.05, which means that that model did not capture the actual number of co-occurrences. And it is blue if that model did capture the actual number of co-occurrences. Now we can see here in the upper left that the null variant has a large number of blue tiles, which tells us that when we only control for the prevalence, those models can still approximate the number of co-occurrences fairly well, which means that, and I think that provides evidence that we really need to control or the importance of controlling for that prevalence when we identify pathogen pairs, like what we did in the previous section. But what we also see is that when we count up the number of pathogen pairs that are approximated correctly by blue tiles, we see there is a lot less compared to the 3 specialized variants, whether it's the community, environment, or full variant. And this is especially true with the environment only variant in the context of the newborn model, which means that in an out of sample accuracy context, the newborn model does much better when we only incorporate the shared response to the environment. In the infant model, we see a similar pattern where the null variant does worse compared to the 3 specialized variants. But what we actually see here is that the community variant does much better compared to the variants containing only the environment information and the full information. So we see a flip in the infant model where that residual correlations between pathogens becomes very important. And now what these figures rely on is comparing a p-value to some arbitrary threshold that I selected, 0.05. But I think the more valuable information can come from the shift in that p-value when we compare the specialized variant to the null variant. And so what we'll do is we'll take the difference between the p-value in that specialized variant and the p-value in that null variant. And what that means is that if that value, that difference is positive, then the estimated range of co-occurrences in that specialized variant shifted closer to the actual observed number of co-occurrences. And to simplify, you could say it becomes more accurate. And based on how well that specialized variant does compared to the null variant, we'll classify it into one of 4 categories. And then we will also, for a pathogen pair and variant combination where the null model alone is sufficient, we're only going to consider that because that's the simplest model that only factors in prevalence. And so if that model is sufficient, that trumps all other variants. And so what we'll do is we'll compare the shift in that p-value for between the community variant and the null variant, and the environment variant and the null variant along the horizontal axis on the left and right figures respectively. And then we will compare the shift between the null variant and the full variant along the vertical axis in both plots. And for example, what we see here, if I call your attention to EEC and ETEC, this is a pathogen pair that falls along the positive axis, along the positive value along the horizontal axis, but it falls close.
Is Connor still on the call?
Not sure.
Houston, we have a problem.
Yeah, I think we lost him.
Can somebody text him?
Let's just wait and see if he can log back in.
I can text
I'll reach
you.
out to him.
Oh, did something happen? Did I cut out?
Yeah, you did.
Yes, we lost you for 30 seconds or a minute.
Okay, where exactly did you lose me?
You
Oh, I
were
know.
just about to explain the EAC, ETEC, the shift in the p-values for EAC and ETEC in the environment
Oh.
model.
Okay, all right, let me go back to that then. Zoom issues. All right, are we all set? Can everyone see me and hear me and everything?
No, we lost your screen, at least on my end. So if you could
Yeah,
share.
no screen share.
Oh, okay, all right. Let me give me 1 second.
This is the portion of the defense where you demonstrate grace under pressure, Connor. No worries. You're doing great.
Thank you. Thank you. All right. Are we all set? Am I showing the right screen?
Yep.
All right. Beautiful. All right. So I was just starting to explain E-Tech EAC.
Yes.
All right. Okay. So, I want to pull your attention to EAC E-Tech, where there is a positive, a shift in the positive direction along the horizontal axis. And what that means is that when assessing the accuracy on the testing data, that for that pathogen pair, the model that only incorporates the environmental information does much better than the model that only incorporates or that incorporates the environment information plus those residual correlations between pathogens, because this pathogen pair falls along the 0 intercept on the vertical axis. Versus for the 8 pathogen pairs outlined here in green, those pathogen pairs fall along the positive direction in the vertical axis, but close to 0 on the horizontal axis, which means the model that controls for the environment as well as incorporates those residual correlations between pathogens does much better in out-of-sample accuracy than models that only include that environmental information. So that means that for these pathogen pairs, even when we control for that environment, there's still something left, possibly a biotic interaction between pathogens that is needed to approximate their co-occurrence. And what this shows is how we can compare a shared response to the environment versus perhaps there's something that's needed left over, which would be incorporating a biotic relationship between those pathogens. So in conclusion, with the joint species distribution model, we can compare the correlation between pathogens, which might represent a biotic relationship between those pathogens, and or a shared response to the environment versus a shared response to similar or opposing environmental niches. We find that when we analyze the posterior distribution sampled on that training data, we find that the strength of that shared response to the environment is much stronger than the residual correlations between pathogens, which means that when we look at those posterior, the environment information is very important compared to potential biotic relationships between them. But what we find is that when we look at the out-of-sample accuracy or the testing accuracy, it depends on the age of the individual, but some pathogen pairs are approximated more adequately with incorporating the environment information versus community information, or some pathogen pairs need to control for the environment plus include a residual correlation between them, representing some biotic interaction. And so what do these two models tell us? Well, it's not going to be a one-to-one match, because if you remember in the configuration model, we are only including up to the first year of life, or only including data collected during the first year of life. And we only look at the Dhaka, Bangladesh site. Versus in the JSDM, we expand out to 15 months, and we include all 8 study site locations, and we only use the MALI-D data set. But I think what we can look at is a qualitative intersection between the two models, where we can identify pathogen pairs that appear outside their individual prevalence. And then we can characterize those pathogen pairs as a shared response to the environment versus a correlation between those pathogens. And what I did here is to exemplify that is I took the 8 pathogen pairs that from the infant model in the JSDM, where these pathogen pairs, when even after we controlled for the environment, they still had some residual correlation between those pathogens, indicating a possible biotic relationship. And then I looked at the pathogens that were in the top 20 pathogen pairs from the configuration model, ranked on based on their discordance. And pathogens that appear in both sets are those that are bold and italicized. And then pathogen pairs that appear in both sets, those edges are outlined in red and bold. And what those pairs tell us is that those pathogen pairs appear more or less than we would expect given their individual prevalence. And when we controlled for the environment, there was still a small amount of residual correlation between pathogens that still had to be accounted for, which could be a potential biotic relationship between them. And then the dashed lines between pathogens indicate pathogen pairs that we found in the configuration model, but when we control for the environment in the JSDM, that signal goes away. And then the blue edges represent pathogen pairs that had some residual correlation when we controlled for the environment, but when we control based on the prevalence, that signal or that discordance from what we would just expect based on their prevalence is not as strong. And so this would, in closing, well, let me walk that back. In closing, what we can find is we can identify pathogen pairs that appear more or less than we would expect, given their individual participant, given their individual prevalence, and then also the impact on that participant. And then we can characterize pathogen pairs as a shared response to the environment versus a residual correlation between pathogens. And then what I think these two models can really show us is, as I did previously, is you can use them in tandem as a sort of pipeline to identify pathogen pairs, such as the 4 that we identified previously, that could be ideal targets for further investigation in understanding how co-infections work either in a synergistic or antagonistic relationship, and how those relationships work in the context of diarrheal disease. And so thank you very much for coming here today. That's all I have for you. Thank you.
Well
Thank you
done,
so much,
Connor.
Connor. Go ahead, John.
Yeah, sure. If you want, Laurent, I think I can see the chat box as well as raised hands for questions. So, and Maggie's got a question already. So, you know, Connor, I just want to say in the big picture, I think what you definitely demonstrate is that these big data sets from an epidemiologic purpose can really be great. And these big observational data sets can be really great for generating, you know, hypothesis generation for further experimentation. So I really liked how you ended with that sort of point. That was awesome. Thanks. Maggie, you have a question. Go ahead.
Yeah, so nice job, Connor. I just have a question. If you can go back to that, you had the correlation matrices in color with blue and whatever for the testing data. There, that one.
This one
Go
here?
forward. No, go forward.
This one, yeah.
That one. This is just my not understanding what you're showing me, but why aren't the main diagonals all blue? Isn't that just comparing something with itself?
Yeah. So what that means is, yeah, that is correct. So what that means is that the comparing something with itself, yeah, that is correct. So that means that for those pathogens.
Oh, wait a minute. They are all blue. Oh, never mind.
No, there is.
It looked like there were some gray boxes in there, but in looking at them closer, maybe they are all blue.
Yeah, I had that
Some
trouble
are
too,
yellow.
Maggie, in terms of looking at I think the lines between the squares are pretty thick. So you see this visual thing, but there are a couple where
They're
where
yellow.
out above the p-value, they're yellow, right? You know, so that maybe, Connor, I think you were going to address that. So go for it if you've got, you know, if you want.
Yeah, and that's a good question, because you are right. This would mean that for approximating basically that's the individual prevalence for that pathogen, and that's a really good question. I think I'd have to look into that further, but what that means is that for that individual pathogen, when we only look, because this is a multiple regression. And what that is it's, you know, it's layered regressions on top of each other. I would say that for that pathogen, these individual regressions, there's evidence that they are just not very good at, perhaps for that individual pathogen, whether we include control for the variance, the correlation between pathogens, or all those environmental predictors, right? So I think because it's sampling from multivariate normal, and then that standard deviation would just be with itself. I think that's fine in saying, well, the correlation with itself is not a good predictor for that pathogen. That can make sense. And then I think for the environment context, what that means is that for that individual pathogen, the environmental predictors we provide are just not good enough. So I think this could provide, kind of a good additional filter to say, okay, it should be able to predict those individual pathogens, but perhaps for these variants, it's just for that individual pathogen is not good. So I think that might provide weight to the idea that, well, maybe for then any pathogen pair where that pathogen occurs, maybe then we don't have as much faith in that pathogen pair being approximated correctly. So, but yeah, you're right. I think they should be able to predict the same, but I think I'd have to investigate that further to see why exactly that's not happening for that pathogen.
If anybody wants to type a question into the chat box too, in case we're missing raised hands, go ahead. You can do that and I can read it out. Ben, go ahead. I see.
Yeah.
I know you're applauding. But do you want to add something?
Yeah, just a couple of quick questions. So first off, terrific work. I mean, it's so much work and it's so interesting. So great job. I had just a couple of quick questions. I think it was slide 26 early on when you were talking about those pathogen pairs that appear to be more highly found increased in pairs more than expected just based on prevalence alone in diarrhea.
Okay.
Yep. That was
This
it.
one?
No. Go back 26, I think it was. Yeah, the bacteria plus virus pairs, bacteria, but those in the red boxes. So
These ones, okay.
Yeah. Just out of curiosity, so I don't remember if you had mentioned this or stated this. Is this were you looking in any way at quantity of pathogen detected, or was this all just qualitative yes, no, it's there or not? So that's one question. And then the other question I had was, in your environmental modeling, was one of the components of that, did you account for seasonality? Because some of these pathogens occur in specific seasons versus others. So I don't know if that was one of the either environmental or other factors that would have been incorporated into that framework.
Yeah. So let me actually, if I can, answer your second question first, because that's the shorter answer.
Okay.
And we did incorporate wet season in there, which is I went through and looked for the wet seasons for all these different sites, and then assigned them as wet or dry season. And that was a very important factor. And then we also saw that, I didn't show it here, but we also looked at what the sign of that response was for each of those selected models. And we did see the same we would expect based on the literature for rotavirus and ETEC, to bring up my earlier example. And so it did incorporate that, but whether we went to a finer resolution beyond wet or dry season, we didn't do that. I only included that as the only representation of calendar time. And then your first question is, yes, that is an excellent point. I think quantitative information is important, right? What we did in this model, and for essentially both models, the JSDM and the configuration, is we used a binary yes or no, are they present, based on if there was a detection via qPCR. Now, I'm very aware that for qPCR, it's very important, right? Do we know if that pathogen, we could still potentially get a signal if that pathogen was cleared. Also, the amount of a qPCR, the amount of DNA found in a sample can determine whether that participant likely displayed symptoms of the disease. And the reason we didn't incorporate it is because it becomes very difficult to define for a pathogen what exactly is an edge in the bipartite network in that case, because this can change based on the age of the individual, this can change based on location, and this can change based on study, right? So this, and so I think it's very difficult. I think it is a thesis project in itself in order to effectively map what the qPCR data is to an effective edge value. And I definitely think that, if as this work further develops, I think that's like the biggest thing to address. But I've been thinking about it for 2.5 years. I haven't quite thought of a good solution for it yet. So we went with just thresholding it because, I know personally that qPCR is very sensitive and very specific. So I think, rather than put in a possibly ill-defined model on top of this model in order to map those values, we went with the threshold because I think it's still reasonable to threshold at this point. But I definitely think it's room for future work.
Yeah, no, I agree. I mean, I think the approach was reasonable. I think that even if it's not incorporated as trying to figure out a way to incorporate up front, I just wonder that might be something just interesting to do, just a quick follow-up on for those identified pairs. Does something emerge? Then you drill down and look at quantity after you establish that there's at least a threshold effect.
Definitely. I 100% agree. And there are methods where once we use these more weighted edges, we can start to use different variants of configuration models. So we can also kind of incorporate that randomization as well in there. So it's really, I got tripped up at the first step, which is how do I define that edge based on the qPCR value? So.
Thank you, Connor. I'm glad to see you still have so much energy before the private of the defense drilling begins. We're over the hour. If there's any last burning quick questions, feel free to raise your hand right now. Otherwise, I suggest anyone that feels comfortable can unmute themselves and just join me in thanking and congratulating Connor for a great presentation. Thank you so much.
Awesome.
Nice job,
Well done,
Connor.
Good job, Connor.
Good
Fine
job,
work.
Connor.
Well done.
Thank you.
We get to hear a little of what's next for you.
Yeah, I will be working. I will be starting in a few weeks as a data scientist for Thermo Fisher Scientific, which is the company that makes a lot of qPCR tests, especially for COVID-19. So I'll be on the team that kind of watches the data as it comes in and makes sure everything's,
And
up
where
to
is that
code.
located?
So I was fortunate. It was gonna be in San Francisco, but they allowed me to start. They allowed me to do it remotely. So I will be. I don't know where I'll end up now, but for now I'm gonna be in Connecticut, just working remotely, and then who knows? Time will tell.
Good luck.
Thank you. Thank you very much. Thank you for coming.
Congratulations, Connor.
Thank you. Thank you.